Okay. Last time you were working with genetics problems, you looked at simple dominant recessive types of genetics. And we're going to follow a trait that actually does not follow Mendel's rules. In this situation, I want to take a look at following flower color. And so the gene is going to be the color gene, but in this case we see a very unusual thing. We see that sometimes in a certain flower, perhaps called the snapdragon, that if a red flower is crossed with a white flower, we get a pink flower. Now, that's very different than a different type of situation in other flowers where you might get a phenotype where the flowers might be red or white, or we might get flowers that are red and white both. So we've got two very different types of inheritance here. One is called incomplete dominance, where neither trait is really dominant, and both end up kind of blending into a new phenotype. And that's actually the situation here, where the pink isn't red, it's not white, it's a brand new phenotype. It was not powerful enough, the pink and the white and the red were not powerful enough to overpower or both be seen. So this is what we call incomplete dominance. So to represent this, if we have a red flower, and if we're going to take a look at pure, if we were going to look at the genotypes of pure plants, we need to use a very different way to represent that symbolically. And we have to use a C for the color gene. And the color gene in this case, if a plant is red, one parent gives a color gene that carries red, and another parent gives a color gene that carries red. If a plant is white, the color gene comes with a white allele, and the other parent carries a gene that carries the white allele. However, we see something different in pink. In a pink plant, one parent donates the red allele, and the other parent donates a white allele. This is called incomplete dominance. And so what I would like to do is follow the genetics of a red flowered plant crossed with a pink flowered plant. And when we take a look at that, we're going to use a Punnett square. And we're going to say, okay, well, this red plant can create sex cells that carry the color gene for red or the color gene for red. The pink plant will carry the color gene for red, but then the other gene it will carry is white. And so this plant, if this sex cell is fertilized by that sex cell, what color is that one going to be? It's carrying two red alleles on its color genes. It's going to be red. Good. If we take a look at this one, color gene for red, color gene for red, it too is going to be red. If this sex cell is fertilized by this sex cell, what's it going to look like? It's going to be pink. Excellent. And if this allele, this sex cell carrying the red allele, is fertilized by this sex cell carrying the white allele, what color is it going to be? It is going to be white. Is there any chance? Oh, wait a minute, it's not going to be white. If it's carrying a red and a white allele, it's going to be pink. So, genotypically and phenotypically, we don't have those funky ratios like we had before. We have to actually spell it out and say, what is our ratio of red to pink to white? Now, I could have said white to pink to red. You usually put the incomplete one in the middle. How many red do I have? I have 2. 2 red to 2 pink to no white. So I have 0 white. So that's how you handle something referred to as, in this case, incomplete dominance. Incomplete dominance. But now, I'm going to take a look at the problem over on the right-hand side of the board. Just going to erase this for now. Over here, we see a similar type of way to write it, in that we have the color gene. We're going to use the red alleles, the white alleles, but in this case, phenotypically, the heterozygote is going to express both traits. This is a situation where both red and white are equally dominant. We call that codominance. So we actually work the Punnett squares an awful lot alike. So I'm going to take a red plant for this particular example, and I would like to cross a red plant with a white plant. Now I've just given you their phenotype. This is going to be the genotype of the white plant, and that is the genotype of the red plant. If I create a Punnett square, it doesn't matter which one goes on which side, and then I represent a cross of the alleles, well, I start to see something interesting happening here. They all contain the same combination. What are all four of these offspring going to look like? Red and white. If it's codominance, they're all going to have red and white striped petals. So how many, when we take a look at our ratio, how many red and white, and then how many white? We have 0 red, 4 red and white, and 0 white. We have a ratio of 0 to 4 to 0, both genotypically and phenotypically. So that's how you work codominance. So now you and your partner are going to have some problems to work through on your own that take a look at incomplete and codominance.